Hi, welcome to Biology Videos at TDA. So I'm going to be starting Year 13 work now. So this is the OCR Chapter 13, Neuronal Communication. And we'll be looking a little bit at homeostasis, and that'll go into the next topic. Just a heads up for submission of this, at the end there'll be a little summary. But what I want you to do, in short, is make notes, do the activities, do the questions, and then either take a photo of or upload it via a PDF, scan it, however you want to do that. But a photo on your phone will be fine of the work that you've done with your score. So just a heads up. So if you work through the lesson, aim to do a sheet of A4 and then upload that to me. Okay, so we're doing section 13.1 and point 2, there's a little joke here, let's see if you can get that. And we're going to be looking at neurons in a bit more detail in this lesson. So objectives of this is just to kind of recap from GCSE and introduce this topic to you, and understand why we need these communication systems in multicellular organisms, how do we do that communication from cell to cell, and therefore the structure and function of the different neurons, particularly sensory, relay and motor neurons, going into detail into the subcellular structures as well. Okay, so homeostasis in general, a reminder from GCSE, it's the ability for the body to maintain internal conditions, to keep the body the same. So some examples of ones in the body that we have are blood glucose concentration, temperature, water potential, cell pH, and there's some externally that will then affect internal, such as humidity, that will affect how much we sweat, as well as temperature. Light intensity is a bit more important for plants, but also for our sight as well, and then sounds. So why have we got this communication between them? Well, some of the main reasons are that cells have become specialised, so they do one particular function. So one example of that is a red blood cell. So for a red blood cell to do its function of carrying oxygen around the body, it needs other cells to communicate, such as to the hemopoietic cells, to create red blood cells and then have the nucleus removed, because then they then can't do their own functions. And a lot of the tissues inside the body, they don't work on their own, they'll work with other tissues, such as the cardiovascular system, so the red blood cells themselves working with the heart tissue, the heart organ, and then all the vessels at the same time. So plants themselves will need to be able to have some kind of communication. We've looked at previously communication regarding pathogens, but such as pollination is another example. So the temperature and the light of the surroundings in the different seasons is going to need to be detected by the plant and then cause pollinations at particular times. And then finally, we need to communicate between organs. So another example you should be familiar with is blood glucose concentration. So we've got the pancreas, the liver and the duodenum, that's the beginning bit of the small intestine. They all work together to control that glucose concentration. So what is cell signalling? You'll have seen this term already before, but cell signalling is the communication between cells. And a new term you might not have seen before is ligand. Now in chemistry there's a more complicated explanation regarding different parts of the molecule, but in short for biology we're looking at a molecule that binds to a receptor and then causes a response. So it makes a change. That's what we have as a ligand. You'll hear the phrase ligand-gated channel, and that means some kind of channel, normally a protein, a channel protein, a sodium protein, that will open up when something binds to it, an example of sodium. And the usefulness of a ligand is that they'll bind to receptors, specific shapes. So if we have another cell that doesn't have those receptors, it won't cause a change in that cell. So this is really useful for things like hormones that will travel all around the body, and we don't want them acting on all of the cells, just particular target organs. Now this can be locally, so this could be from a cell to the adjacent cell, or it could be across the entire body, from such as the pituitary to the kidneys. So that's going to cause ADH to change how permeable the distal convoluted tubule is to water. And in plants they'll do the same. So one part of the plant will detect the light, and then that will cause auxin to grow. If you remember, that's on the shaded side of the shoot. So that kind of is just an introduction of 13.1. That's why I've done it quite quickly. This is the bit where we'll be learning a bit more. Now again, this is nice, it's built on GCSE, but we go into more detail, and the explanation is going into more detail. So when we have the body and some kind of response, they'll always go through some kind of system, and that will go from a stimulus. So the stimulus is the change in the external environment. Now it can be internal as well. So at GCSE we learn it is only external, but at A-level we know really in more detail it's internal. So it could be baroreceptors detecting pressure, chemoreceptors detecting pH, that kind of thing. We're going to process it in some way, and then we're going to respond. And normally in homeostasis it's to bring it down. But if it's something like positive feedback loops, such as contractions in a female during birth, we might actually increase it more and more and more and more until you get to an end goal. So let's look at the neuron cells themselves. So here I'm going to I've got the sensory and the motor neuron. On the next slide I'll show you a relay as well, but it kind of fit here. So you should be relatively familiar with the general shape, but I will recap. So we have the cell body, which is the cell membrane with the cytoplasm inside, and the normal things of a cell you would expect to be in it, except they have higher amounts of endoplasmic reticulum and mitochondria, because they're going to be needed to produce neurotransmitter, and lots of energy is going to be needed by these cells to do various different things. We've got the axon, and the definition of the axon. So it's a singular elongated, it's like a tube that transmit away from the body. So this section here is referred to the axon, this section here is referred to as the dendron. If you call it an axon in general, the whole thing, you'll be okay. But just to be aware, there are a difference between the two. They're normally about 1 micrometer in diameter, so just be aware of that. But they can change in size, and the larger the diameter, the faster the impulse. But we'll cover impulses in a later topic. You've also got the dendrons, so the section before the cell body, and then you've got the dendrites that then split into the smaller sections. So the dendrites are the smaller bits, and there can be many of them. Some neurons can have up to 1,000 dendrites and 1,000 therefore connections with other cells. So one cell can affect that of another 1,000 cells, and that kind of multiply. Okay, so let's look at the three types then. So we've got the sensory neuron. You need to be aware that the cell body is segmented away from the axon. You've got the motor neuron, in which the cell body is at one end of the axon, and the relay neuron, in which the cell body is at the centre of the axon. Okay, so relay neuron just takes information and passes it immediately straight on. We're going to go into a bit more detail further on about myelin sheath node of Rumbier, so we'll look at that later on. But a reminder of this process that we go through: we have a receptor, which is not seen here, so a receptor could be a Peninsula corpuscle, or it could be a rod or a cone cell, it could be a baroreceptor, and that's going to detect some kind of stimulus. It's going to transduce it, so it's going to change it from whatever form it is into an electrical impulse, and that will then arrive at a sensory neuron. So once we get to the sensory neuron, depending on the type of the response, but if it's a reflex action, such as a knee-jerk reaction, we're going to then go straight to the relay neuron where the information is going to go in. It's going to be passed straight on then to a motor neuron, and that's going to then go and end up at the effector cell. So an example, such as the knee-jerk, well, the stimulus is going to be stretch of the tendons and ligaments in the knee joint, just below the patella, this is the kneecap. That's going to cause an electrical impulse, that's going to go down the sensory. The relay neuron is found in your spinal cord, that's going to go back down to a motor neuron, which will go back down towards the leg, and that will cause the quadriceps, which is the effector, the quadricep muscles to contract, which causes you to straighten your leg, if your body thinks your knee is overstretched. That's why we have the knee-jerk reflex. So you need to know the three different types, and you need to be able to tell us this process that goes along. So your first task, you need to do this off by heart. So pause the video, and this can make it part of your notes. Draw and label 3 types of neuron. Okay, so now we're going to look at the nodes and the myelin sheath in a bit more detail. This is where kind of the A-level kicks in a bit more from the GCSE. So we've got an electron micrograph. A reminder, what type is this? So you can have a scanning or a transmission. This is like a transmission electron micrograph because it is two-dimensional. The colour has been added on by a computer. We don't see colour from them. And we can see the magnification of about 25,000 here. And you can see the myelin sheath goes around the axon, which is in the centre, and I've got another example here which is not in colour. Now the Schwann cells, we can look at those in more detail in a second, they wrap around more than 20 times, and there's another cell sort of similar to neuron cells that will help this get here. And they provide an electrically insulating layer, so they're going to stop electrical activity moving too far away, and we'll come to the action potentials later on. And that will go at about 100 meters per second, and we will do sort of practicals around this, and we can detect how fast reaction time, and that's why things like in 100-meter sprint races, if people go too fast even after the gun, we know reaction time is only around 100 meters per second. So if someone does it at 500 meters per second, if that's their reaction, we know that person then has anticipated the gun. But a non-myelinated neuron, a non-myelinated neuron is only about 1 meters per second, so you can see the massive advantage of having this myelination. It also allows us to require less energy because we have to have less depolarization in the action potential, which means less sodium potassium, which is moved. We'll get to that in a later lesson. But the key point is less ATP is needed because we have this myelination. So it also saves energy, which is a really useful thing to have. So we'll look at the node of Ranvier here. Okay, so the node of Ranvier is the gap between the myelin sheath, okay, which are made of these Schwann cells. Now what this does, and you should be really careful with your wording here, is this allows for a local current between nodes of Ranvier to occur. So the key point to write down here is it creates a local current. Okay, you cannot say in the exam it jumps from node to node. If you do, you won't be marked wrong, but it'll be ignored, and I'll show you an example of this in a second. And what this does is it means that the current passes from node to node more quickly, so that's why it speeds it up. So the key point to make is it creates a local current. And you can kind of see it from this image here, which I'll zoom in on. So we've got the myelin sheath here. So we've got this positive and negative, and we'll get to learning what that means in a later lesson. But we've got this local current that occurs, which means the rate of transmission of the charge is much more quickly, whereas when we have continuous conduction, we have no myelin sheath, is much more slowly. And we refer to this as saltatory conduction, and we're going to kind of assume saltatory conduction from now on. Now you will get some questions that will compare the two together. So you've got continuous conduction, which doesn't have myelin sheath, and saltatory conduction, which does. Okay, so that brings us to some exam questions and some practice on that. So here's the first question. So mollusks have unmyelinated neurons. Saltatory conduction cannot occur in these neurons. Why is the transmission along the action potentials slower in the absence of saltatory conduction? Have a go. Pause the video and have a go. Okay, so you should have put it creates a local current, and you could put a shorter current with no node of Ranvier. And you can always write the opposite. So if you're saying that in the mollusks, in this question I believe, yeah, in the mollusks, they have no nodes of Ranvier, so it means they're slower. If you set it the other way around, you'll normally be okay. And you can see here in the guidance, ignore the reference to jumping between the nodes. You cannot say, well, the electrical current jumps between the nodes. It won't get you the marks. But by saying it creates a local current, does. Okay? Something else that I forgot to mention in regards to nerve transmission is, this is quite a common question that you see come up, is certain shellfish and other organisms contain neurotoxins. And one of the ways that they work is they block these channels. And in here there's some sodium-potassium channels. And another example that I've seen in exam question before is TTX, which is tetrodotoxin, which is in Japanese pufferfish. You might have seen or heard of examples of where they have to cook the pufferfish in a particular way. And one of the ways they work is they block the voltage-gated channels, the ligand-gated channels, in some of the nodes of Ranvier, which means essentially you're not going to pass on electrical impulses. Now, if that's of something important, like your breathing and your heart rate, that can be very dangerous. You also have local anaesthetics with drugs. An example is Procaine or Novocaine, which can produce anaesthesia in the skin. So when you might get something like a dentistry injection, or if you had a cut and you need to have sewing up, they might inject some of this local anaesthetic. And one of the ways that they work is they block these channels from propagating, from doing action potentials. So this is kind of question you might come up. You might see an example. You need to apply, well, how do they work and why does that affect the body? Well, you would say that if you block these channels, you can't create your local currents, and therefore you can't pass an action potential down the axon. Okay, let's have a go at another question. Well, before we do, another example of where this can be applied in pathophysiology, so disease, is multiple sclerosis. So this is a computer outlet model of multiple sclerosis. This is the breaking down of the myelin sheath around the outside of the axon. And about 1 in 100,000 people get it. It's autoimmune disease, which means it's your body attacking itself. We think it's a combination of genetics and viruses, like a lot of autoimmune diseases. People can have genetics, but the reason they actually come around might be induced by a virus. And viruses, we're learning, such as coronavirus, COVID-19, they create changes in your DNA, and that might be the reason why, but we don't fully understand it. And people are most likely to be diagnosed from 20 and 40 of this, but it's a pretty rare condition. But you could kind of, if I was to give you a question of, well, how might this affect a person? Well, you would go into detail now and say, therefore the rate of transmission is going to be much more slowly. So therefore the control over a person is going to be much more slowly. and they're not going to be able to respond to stimulus, which can lead to that kind of jerking or slow or the complete inability for a person to do certain things or parts of the body, and that leads to muscle wastage and kind of wastage around the body. Okay, let's have a go now. Let's finish off this lesson with some more questions. So which one is a sensory neuron? And it was D, because the cell body is away from the axon. Next question. Outline the ways in which the structures of a sensory neuron and a motor neuron are similar. Okay, you need to be very careful with your spelling because there's similar words here and using appropriate technical terms. Okay, pause the video and have a look. Okay, something really important in this question, I didn't highlight it, I wanted to see if you spotted it, but in bold here it says they are similar, so we're only looking at ways in which they are the same. If you start talking about the difference in the position of the cell body, you get no marks for that because it's similar. So let's have a look how they're similar. So, they both have dendrites, they both have axons, they both have a cell body with a nucleus, and you could put named organelles such as endoplasmic reticulum or mitochondria in there. They both have a myelin sheath covered in Schwann cells and therefore nodes of Rumbier. They both have voltage gated, or these ligand gated sodium channels. We'll come to this step number 5 later on. Okay, let's have a look at the next question. So name this type of neuron and state the function of Y. Okay, so as we can see, it was a motor neuron because in the position of the cell body. And then what is the function of Y? Well, that's the myelin sheath. You need to tell us what it is. You need to tell us what it does, the function. So it does saltatory conduction, and it does that by increasing the speed or the rate of the transmission of the action potential. And it also insulates the axon. Relatively simple question. Next one. Multiple sclerosis MS is an autoimmune disease that damages the nervous system. Suggest how the immune system causes damage to the nervous system. So this links you back to chapter 12. Okay, so it's really important that we think about the immune system with this answer, because it hasn't asked for any of the structure or function of Schwann cells at the moment. It's looking at how does it cause damage to the nervous system. So, why do we have damage of an autoimmune disease? Well, why do we attack parts of our own body? Well, the number one key point is it must have antigens inside the cell. So there must be the antigens on the neurons or the Schwann cells or the myelin sheath which activate the immune system. It could be doing it wrongly, but it does activate it. That then causes us to produce antibodies. How do we fight disease? Well, it's by antibodies, which are then going to attack or attach to the Schwann cells. That causes phagocytes, neutrophils, macrophages or killer T cells, all these kind of words we looked at before, to break down or attack or damage these Schwann cells. Okay, and the final question of the day, we've got an image here of a neuron that has been affected by multiple sclerosis, and we've got some annotations. So you've got the purple, this is the axon, you've got the light blue, so you've got the Schwann cells around the outside. I know you can't see it that well, so I'm trying to highlight to you. And then the black is trying to show that these have a myelin sheath. So the key point is, the exam board are trying to show you that these cells have a myelin sheath, this one doesn't have a myelin sheath. So using the information from figure 5, which is this one, what can you conclude about how multiple sclerosis causes a reduction in the speed of a nerve impulse? Okay, so how does it slow it down? Well, one of the ways it slows it down is there's no or there's dead Schwann cells. So therefore you've got less electrical insulation on the neuron. So therefore you have a lack of saltatory conduction. You've got the inability to have the local current. So you're able to describe the saltatory conduction here as part of your answers. Okay? Right, thank you very much. That's today's lesson. So to submit this, can you please take a picture of what you have done? Please don't email me. Please upload it directly to show my homework, and I'll have a look through and give you comments. Aim for one sheet of paper. If you've done more than one sheet, as I've been talking through, that's fine, but you only need to upload one of them. You can upload more to show me it, and I will read it if you've done that. And then can you just give yourself a mark, and it is out of 12. So just put that on there, 10 out of 12, or whatever you might get. Thank you very much, and I'll upload another lesson later on this week. Thank you.